Thank you so much for the invitation, and it is a great honor to be here today with all of you and to be bringing you some of the research that we're doing at Lethbridge Polytechnic. As you can see, this is a multi-project in terms of collaboration, so we have other partners that are supporting, helping, managing this project that I'm bringing to you today. So we have Nature Conservancy of Canada supporting us, and also as a collaborator, there is Athabasca University, University of Calgary, and our project is being funded by Alberta Innovates. I would like to start my talk today with a phrase that I believe resonates a lot with everyone in this room. So this phrase from William Singer starts saying, warriors still exist. So it is a warrior's duty to protect the land and to know the land and all its stories. At the same time, now we have to protect the land because the land protects the people. And this resonates deeply with the work that we do, and it also summarizes everything that is a goal for everyone in this room, I believe. So we want to protect the vegetation, the land, the grasslands, and also keep it as much as possible in an intact stage for the future generations. Grasslands, which is the lands that we're focusing in our research, are amazing. I mean, in terms of services, they go from ecological services to agricultural services to all kinds of small actions, going from even providing the first flowers that pollinators see early in spring. And it is not a surprise for us to know that grasslands are in danger. So they are actually disappearing at a faster rate than when we can recover them. This is a global problem. It's not only in Alberta or in the prairies or United States as well. It's everywhere. So they are just disappearing faster than the rate that they are reclaimed. It takes years to recover soil. It takes years to basically bring the land to what it once was. So traditional restoration practices do an excellent work at bringing back some of that restoration approach that we see bringing back the grasslands. So we are talking about grazing management, we're talking about soil amendments, erosion control, but in general, there is a big focus on the above ground restoration. So controlling the invasives, prescribed burning. We've been talking about these methods, and they are effective and they are really practical, but I believe that we're missing part of the puzzle. I believe that we're missing the microbial community. We're missing looking deeply at the soil and understanding that the biology in the soil will reflect what you see above ground. So my research is aiming to bring innovative practices that somehow could accelerate the traditional practices. Things like microbial mapping, tools like microbial inoculation, also looking at, for example, precision restoration with drones, because when you have eyes from the skies, it means you don't have to set foot on the ground, and that can mean a lot depending on the location and the vulnerability of the plant species that you have in that area. We're also talking about precision soil testing. We know that there are huge differences in soil from pH to electrical conductivity to nutrient gradients from a location to the next, and sometimes it's hard to put that together into a single sample. So precision soil sampling is also important, and together with that, so having that genetic resilience. So also understanding that seeds, they carry the traits of the area that they were formed, that they developed. So understanding and gathering seed from locations that will reflect the weather that these future plants will have to endure. So in my presentation, I will focus first talking about the microbial community, that biology in the soil. When we talk about microbial mapping, we answer the question, who lives there? Who lives here in this soil? And microbial communities are very complex and they are very dynamic. Sometimes they tend to work like a well-oiled engine where all the parts complement each other, help each other, reciprocate each other, creating what we call a healthy soil. But other times, when we see disturbances, when we see activities that are either anthropogenic or natural, climate change impact can shift these microbial communities, and that effect translates to what we see above ground. So some of these anthropogenic disturbances that we see more often in grasslands are overgrazing. We also see mining, climate change, agricultural impacts. And at the end of the day, when we go to our sites, the question is usually, where did the soil go? So getting a soil sample in some of our sites was actually quite challenging, and I will talk about my sites in a couple of minutes here, but this is one of the pictures that you can see that we're trying to take a soil sample, and basically we just carry rocks back home and then sieve them and got some of that soil. But it is quite challenging to restore that soil once that is lost. So microbial mapping in my project brings also a dynamic that pretends to be more holistic than previous research. Most of the research that talks about microbes tends to focus either in bacterial communities alone or in fungal communities. We're trying to see a holistic approach when we talk about microbial community as a whole, because they are not separate, they are depending on each other, and they interact all the time. So we're looking at both fungi and bacteria in the soil. My focus in the lab that I manage at Lethbridge Polytechnic focuses mostly on beneficial fungi. And you probably have heard before about the symbiosis between plants and mycorrhizal fungi. Mycorrhizal fungi are basically the friends of plants for millions of years now on Earth. So they associate with over 80% of the plants in the planet, and from North Pole to South Pole. So everywhere you can find them in the globe. And what happens with mycorrhizal fungi in this association is that roots can reach just so far for water and nutrients, but then this extensive network of hyphae, which is the living body of the fungi, extends outwards and far away from the roots, exploring that soil, and then transferring water and nutrients that are too far for the roots to reach. Oops, sorry. And then they will basically transfer slowly those nutrients and the water towards the root zone and towards the plant. The plant, in return, provides carbohydrates for the fungi to live. So mycorrhiza is an obligatory symbiont. It means it relies on the plants for survival, and the plants can choose whether or not they let mycorrhiza in if it's beneficial for the plant in place and establish this functional symbiosis. So you can see in the conditions that grasslands are, this could be a key component for success and restoration of these ecosystems. Our project spans over 4 years, so we're talking about 2024 starting last year and goes all the way to 2028, again funded by Alberta Innovates and involving multiple organizations and collaborators that are supporting these projects. Our objectives are mapping the microbial communities that are present in disturbed areas that we carefully selected in the Crossness Pass, and then after we understand what's there, also trying to see whether inoculants, so mycorrhizal amendments, commercial amendments that are available could be utilized to speed up restoration. At the same time, we will monitor over time these sites and the changes that they could be that we could see over time as a result of the changes that we introduce with the inoculation. So these are our sampling sites. You can see this is at the Crossness Pass, and we have Site 1 west of Leach Collieries, Site 2 east of Leach Collieries, and then we also took samples of this topsoil pile. So the topsoil pile has been there in the property for decades. This is Servo 2 NCC property, and the topsoil becomes somehow a source of microbial community that is native to the site. Because it's been there for so many years, it already has a plant community growing all above the topsoil pile, and it is constantly used in restoration projects when soil is not quite abundant in the site that is being restored. Then they use this topsoil pile to supply soil in the areas. So we're assuming that contains somewhat native microbial communities that could be used as well in our restoration approaches. When we look at the disturbances that are affecting still these two sites. They are closely related, of course, with the activity of the leach collieries. This mine was active for over about 10 years, from 1909 to 1919, and it was one of the most ambitious projects in the Crossness Pass. So basically, a little bit of that history. Cattle was herd in the area for many years before the mine was created, because the valley offered shelter, water, and of course abundant grass for the cattle. At the same time, later on, when the mine was opened, then it was one of those ambitious projects. It was the first Canadian-owned mine in the Crossness Pass, and it was supposed to be really big. So despite the limited resources in the area, most of the gravel, most of the stone, the bricks, they came from surrounding areas. There was a railroad that just went across multiple sites, bringing resources to build the mine. So the mine was able to process about 1,000 to 2,000 tons of coal daily. There was also an electrical powerhouse that was built on site. There was a huge triple structure that was also built with the resources from the surrounding areas. So as a result, and if you ever pass by, you can see actually there is a great historical path where you can read and understand more about what happened in this area. And you can see some of the locations and coke ovens that are still present there as a reminder of what was happening so many years ago. So when we look at the sites, site west and east of the mine, what we see here is the result of erosion and also some areas that are resisting to the natural traditional restoration approaches. So I intentionally left in this picture, so this is the site number 1, and then this nice slope right here is very similar, and vegetation is also equivalent. So we have our disturbed site and then a natural equivalent adjacent that we can use as a baseline for our restoration attempts. The same way here, we have site number 2, and just adjacent to it, there is nice green areas that are perfectly undisturbed or that has been recovered over time. NCC has done an excellent work at restoring these areas, managing this property, and we're focusing on these smaller sites that are still somewhat affected and not being recovering fast enough. So a few pictures from the drone imagery. This is site 1. And you can see here it's just right by this empty area. That's where the railroad actually was passing by. So, and the little dots here represent vegetation, so plants that are already there. So one of the challenges of our projects is that we have to work around what's already in place. We can't go and pull plants and then set up our experiment. We have to work with what's there, understand the impact, and then try to incorporate the practices that we're bringing. So the slope in these sites is a big challenge. Site number 1 right here has a very steep 57.4% slope. So literally you can climb there without rocks just literally falling down at every step you take. So we had to get really creative to repopulate this site with plants, and we'll show you how we did it. Also, the fact that we're using a drone allowed us to take multiple pictures and videos of the area. So instead of using a quadrat or staying for hours trying to count and analyze plants, we just fly the drone. It takes about 15 minutes, go back to the college, and then we just look at the images. And in about 15 minutes, we're able to get all this data. So it saves a tremendous amount of time. Site number 2 right here, same idea, sloped as well, a bit less slope than site number 1, so we're looking here at like almost 28% slope, but still quite challenging, and this area is high up in the terrain, so a lot of exposure to wind and erosion. There is also way less soil in this site compared to the other site. So this is sample collection as a mapping idea. We have representative samples, and we're considering here every flag is a point where we took a soil sample for analysis, so for precision soil sampling, and also we took samples for DNA analysis of sequencing of microbial community, and also microbial community analysis using microscopy. So lots of samples were taken, and we were taking into consideration not only the slope, so having the top, the middle, and the bottom of the slope, but also the aspect of the site and some exposure as well, because all these conditions can change how microbial communities are distributed, and also nutrient gradients, moisture, and so on. The plant species that we're focusing on in our research are the blue bunchweed grass, rough fescue, and the little sister of rough fescue, Idaho fescue. So don't let the picture fool you. You know Idaho fescue very well. It just looks really big there, but we know it's just this teeny tiny, beautiful fescue. And it's one of the plants that we found rely the most in mycorrhiza fungi because it's so tiny and the root system is fairly big for such a small plant, but still, depending on the conditions, it can actually get a bit sad with drought and shading and things like that. So mycorrhiza is actually a big friend of Idaho fescue. These plants are important not only because of their ecological significance in the foothills in Alberta, but also because they are grazing-friendly plants, so they are important for agriculture as well. A bit of the sampling preparation in our lab. This is one of my students basically staining the roots of these grasses to look at mycorrhizal colonization in these roots. And we look at spore counts in the soil, so mycorrhizal spores in the soil. We stain roots and look at mycorrhizal structures within the roots of these grasses. We also look at soil, so basically complete soil testing, drone imagery, normalized difference vegetation index in the plants, and also the DNA sequencing for bacteria and fungi. So when it comes to mycorrhiza, in case that you have never had the opportunity to look at it in a microscope, this is how it looks like. So it is basically formed with different structures that you can easily identify once that you know them. We have hyphae, which is the vegetative body of the fungi, and these are internal in the cells of the roots of rough fescue, Idaho fescue, or blue bunchweed grass. We also have arbuscules, which are very ramified structures that are basically the sites where the exchange is happening between the plant and the fungi. That's exactly when the exchange of nutrients and resources occurs. And then the reproductive structures. So right here, those are the mycorrhizal spores. So all fungi produces a spore, so mycorrhiza is not an exception. It's just that they are microscopic and they are released to the soil for proliferation of the fungi. So when it comes to the sites, how much mycorrhiza can we find in 1 gram of soil? Well, we found actually quite a bit. In the sites that we tested, and this is before we did any management, so just looking at what's there before we took action, there was a moderate abundance of mycorrhizal fungi. We found between 40 to 60 spores of mycorrhizal fungi per gram of soil, which is a tremendous amount, and there was also diversity. So in terms of like colors, sizes of the spores, we could tell that there were different species associated with the different plants that were already in the area, which is promising, is good news. At the same time, we found that slope didn't have a tremendous impact in the distribution of mycorrhiza. We were expecting to see, like there is moisture gradients, the same way there should be microbial gradients that answer to that moisture differences with the slope. But the truth is that there is no irrigation in this site, and when we measure moisture, everything was equally dry, with like less than 1% moisture in some samples, so very, very dry. So we're not surprised to see that mycorrhiza was just evenly distributed because, yeah. So a bit of information about DNA sequencing. This is coming from our wizards of the Athabasca University, our Magic DNA masters. And they are basically looking at these microbial communities using primers that are specific for bacteria or fungi. And these graphs, colorful, perhaps a bit complex, but they just answer the question how diverse the microbial community is. And they are showing all the samples from either the 2 sites or the topsoil. So the fungal community analysis is still in progress, so we have only looked at the 2 sites. But for the bacteria, we also have the topsoil represented in green. So when it comes to the results in this area, what we found is that there was actually low diversity in general. So they saw that there were not a whole lot of differences between the sites, perhaps a little higher diversity for the fungi in the west side, but not really very significant. But the most important part was that we expect to find in a healthy soil 1,000 bacterial species, 1,000 fungal species, millions perhaps, and we found way below that. And that shows the degraded nature of the soils. So there needs to be some action in terms of restoring these microbial communities. These colorful graphs, they answer to community composition. So now we're looking not at how many species there are, but who are these species. So which are the families of bacteria or fungi that are present. And the different colors represent percent abundance. So the length of each color is how much we have of that particular microorganism. And what you can see is that there is a lot of difference. So the colors are everywhere. It's a big rainbow. There is a lot of diversity, although there is not a lot of richness, if that makes sense. So we have differences between the sites for both the bacterial and the fungal community, which actually makes these two sites ideal for our research, because we want sites that are contrasting to see if there is an effect of these differences on the treatments that we're introducing. Looking at normalized difference vegetation index, this answers the question, how green is a plant, basically? So we can measure the intensity of the green, which correlates to chlorophyll content, and it tells you how healthy that plant is and how much vegetation is in that area. So we look at the disturbed two sites that we have, and we also looked at the natural equivalent sites that I just showed you in those pictures that are just close by. And of course, we were not surprised to see a huge difference between the natural equivalents for each of the sites compared to the disturbed areas. So that gives us a bit of a baseline to something that we are aiming for. So hopefully at the end of this project, we can see these two bars, which are our disturbed sites, growing and growing and becoming closer and closer to what the natural equivalents are. So for the microbial mapping results, what we can say is that there are microbial differences, so pronounced difference between the experimental sites, and also there are lasting effects that are reflected so far on the vegetation, but also in that soil biology. So that brings me to talk about what I call the probiotic effect. So now that we know that microbial communities have been affected in these sites, perhaps there is a need for these soil probiotics. So the same way that when we have an infection and you take antibiotics, maybe for an ear infection, and your doctor says, well, make sure when you're taking those antibiotics, perhaps take some yogurt along, just to reincorporate the bacteria that's being damaged, or maybe take some probiotics as a capsule. So in that same pattern, we could think about these microbial communities. So the same way that bacteria is to our gut, bacteria coming from probiotics, that will be the same way that fungi, beneficial microorganisms, is to the soil. So it could be justified to introduce microorganisms in these sites because we know that they have been depleted in those areas, and it could be helping the establishment of the communities that we want to establish in terms of plants. So can mycorrhizal fungi help? That's the question. We think that it could, but it hasn't been tested, really. There is a tremendous amount of information of mycorrhizal fungi being used in agricultural settings, in horticultural settings, in greenhouse production, with great success. But at the same time, one size doesn't fit all. And also these commercial amendments have been tested mostly in agricultural crops. So when you take them to grasslands, are the finicky grasses that we're working with happy with these new companions? Maybe they will prefer the ones coming from the native site, the ones that are more closely associated to them day by day. So that justifies our microbial inoculation testing. We need to test this before we actually can tell that it works. Besides, inoculants are very expensive, so it is important to know that they work or not before we can recommend them in grassland restoration. So to do this, we designed a greenhouse experiment. We actually had 4 different microbial inoculants and 2 controls. So here we have mycorrhizal fungal inoculant. Mycos contains 2 mycorrhizal fungi, and then carried in ProMix HP media because it's the media that is commonly used to start plugs and then transfer them to the field. Dynamyco, 2 mycorrhizal fungi as well. This is Green Grow, contains 4 mycorrhizal fungi species and also some beneficial bacteria as well. Then we have the topsoil, collected some topsoil from that native community that's in the field, and we added that as our native microbial community to our treatments. We also had a positive control, that is just the ProMix media, and then we had a negative control, which will be the ProMix media but pasteurized, just to remove any biological action that's coming from that media as well. So our heroes here are definitely my students. They literally baby seed each grass from seed. This seed was collected at Servo 2 property during the summer of 2024. And I literally told my students, gave each a seed and said, this is your baby. Make sure that you baby them, you sing to them every time you come to the lab. You just make sure that they survive. And we literally had like thousands of seeds grown in the lab by these dedicated students of mine. Then we transfer them one at a time. You can see here, they literally look like cat whiskers. That's an Idaho fescue seedling right there. And then the inoculant was added to these root trainers, and then the seedlings were strategically placed right on top of the inoculant. And then we grew over 1,000 grasses in the greenhouse. Of course, the amount didn't transfer completely to the field because we needed testing that implied destroying some of the grasses, like when you're measuring biomass or you're measuring the root colonization, so those were also included for destructive sampling. But many of them, over half of these ones, actually made it to the field. So hopefully you can see these diagrams, but if you don't, the main idea here is the results that we found from the greenhouse experiment. We have on top here rough fescue seedlings, Idaho fescue, blue bunch wheatgrass. We have the treatments, negative control, positive control, topsoil, Dynamyco, Mycos, and also Green Grow. And then we have, in this side is all the plant biomass, so above ground biomass. And then on the other side, we have the root biomass. And we were surprised at first to see that actually these grasses didn't like mycorrhiza very much under greenhouse conditions. In fact, when you look at the biomass values and the bars from the negative control or the positive control, in some cases they were equal to the inoculated ones or they were higher. In this case, for the blue bunch wheatgrass, the topsoil and the green growth treatments did fairly poorly. So we were hoping that topsoil was going to be the star of the show just because it's the native microbial community. And we saw also that it had the highest level of colonization in the roots, which is the next slide. So there is a great affinity for these native microorganisms, but it came at such a cost for the plants that they were barely growing. This is the picture. Here is the topsoil. So considerably smaller plants. And then this is the Green Grow, one of the mycorrhizal amendments. The one that was more complex. So these 2 mycorrhizal amendments are kind of promising, but still not much different from plants without inoculation. So what's going on here? Here is the root colonization levels, and then here is also chlorophyll content in the plants as a measure of plant health. And in this case, we have the expected, so no colonization for negative control or positive control, very low colonization. There was no inoculation here, so that's kind of like a quality control of our experiment. Topsoil colonization by mycorrhiza was one of the highest, and then the other 3 inoculants had small amount of colonization, but still significant. And then when we look at the chlorophyll content, we saw that with the exception of Idaho fescue and Dynamyco, there we saw a significant effect for Mycos and Dynamyco that was better than the controls. So more chlorophyll when they had the mycorrhiza inoculated for the leader Idaho fescue. But for the other grasses, there were not real differences to justify the inoculation in the greenhouse. So then we thought about it and we realized, okay, mycorrhiza is supposed to help the plant. But hey, we're in greenhouse conditions. They have excellent light. They get water every week. So they are happy. They don't need the association. Mycorrhiza is just becoming almost a parasite of these plants in greenhouse conditions. So the question is, once that they transfer to the field, will we see an advantage? Will we see mycorrhiza then shining for those plants, especially the topsoil ones that were somehow the less happy plants? So perhaps there is something in the field when it comes to the stresses that mycorrhiza will help. But in the greenhouse, they were not very happy with the inoculation. So a bit of the field work. It took a large crew of 25 to repopulate these sites in the Crossness Pass. And so this just happened in June this year. So we had plots designed, randomly designed, again considering the slope, and each of the treatment was represented for the two sites, considering a slope and aspect. So basically, at least one of the treatments is at the top, then there's one in the middle, there's one at the bottom of each of these different treatments. You can see here a bit of innovative approaches to get to plant these plugs. So literally, I mentioned the slope on the one side was so big that we couldn't set foot on that hill. So literally, my students got thinking, and one of them said, can we use extension ladders? And I'm like, sure, let's try it. So there we are, one ladder at a time, one person at a time, planting these plugs. We took turns, we rotated, the ladders will move simultaneously left or right, but we got all those plugs in the ground this summer. This is for site number 1, so here, the one that had the very steep 57% slope. For this one, we were able to step in, it was only like 27% slope. But then, again, large crew of people marching together, planting these plugs from top to bottom of the hill. You can see also there is a straw applications. In this case, we didn't want to introduce anything that was biologically active, that was going to change the inoculants, but we wanted to guarantee some level of moisture, so give them a competitive advantage when they go in the field, because there is no irrigation in this area. We literally irrigated with backpacks when we put the plugs, put some straw to protect them from the wind, and to create what we call microsites. So we'll put some rocks around them, a bit of straw, and also a way to mark where the plugs are, because the drone really won't be able to differentiate between our plugs or whatever else was there. So later on, we flag actually each plant individually. So if you're driving by the pass, you might look up and see a pink hill, and it's not flowers, it's actually our flags. But anyways, this is the approach. This is our research that we have done so far. A few take-home messages from year 1 of our research. So we basically have microbial community richness and diversity, NDVI data, and drone imagery highlights the nature of the degradation, the amount of degradation and disturbance, and reinforces the need for innovative approaches to bring these particular two sites closer to what they were once. At the same time, the native grasses relied minimally on the mycorrhizal fungi under greenhouse conditions. We are hoping to see that in the field when there are more challenges like drought or nutrient depletion, perhaps some of these treatments will shine. That remains to be tested and to be seen in the next 2 to 3 years. And so I want to highlight again the importance of looking at this from all the possible angles. So we need to look at the vegetation, we need to look at the microbial communities, we need to look at the soil to get successful and accelerated restoration. So just to close my talk, I wanted to bring just this phrase. So restoration doesn't happen overnight. And we all know this, the endless weeks and summers that we spend out there. So it requires a lot of time and commitment. And for us, the passion starts the journey, the collaboration strengthens the journey, and definitely the innovation will propel it forward. I wouldn't like to leave the stand without thanking our funders, our collaborators. So Alberta Innovates, thank you for the funding. We also have Athabasca University, University of Calgary, NCC, Greenpeace Greenhouses, which was an excellent source of resources and even seeds and information. And you can see it takes a village to restore the site. So this is a really long list of people. If the screen was longer, it will keep going, basically, because we got help from many, many amazing people like you guys.